So at Targovax, we are developing immune activators. And what immune activators do is that they alert the immune system to the presence of a cancer and stimulate an immune response to the tumor itself. And as far as we see it, immune activators will become an important treatment modality in the future cocktail of cancer therapy. For now, they are particularly promising as combination partners with checkpoint inhibitors, but also other drug modalities such as chemotherapy, targeted therapies are relevant for combinations with immune activators. We have 2 immune activator programs in development. Our lead program is an oncolytic virus, ONCOS. This is a genetically modified adenovirus, which is designed to selectively infect and replicate in cancer cells. And with ONCOS, we have 4 ongoing combination trials, all phase 1, phase 2 trials that are currently recruiting patients in Europe and the US. Our second program is a neoantigen vaccine targeting mutant forms of RAS. So RAS is the most frequently mutated oncogene in cancer, and we're able to stimulate an immune response specifically to RAS mutations. Here we recently completed a trial in resected pancreatic cancer, and we have 1 other trial currently ongoing. What's common about these 2 programs is that they both trigger patient-specific immune responses, and there's no need for individualization. So you may have heard of neoantigen vaccines before. They typically need to be individualized to every single patient. Both of our products are off the shelf and available for any solid tumor. Today I'll focus on the oncolytic virus program. So with ONCOS 102 is our lead product. We're also developing next generation versions of the virus. We have completed already a compassionate use program and a phase 1 trial in a monotherapeutic setting. So here we developed our dosing strategy and treatment regimen, and also identified the monotherapeutic activity, as well as which cancers we're targeting. And this has led us to embark on this ongoing program of 4 trials. The 2 dark blue ones in melanoma and mesothelioma we are sponsoring ourselves. The 2 gray ones are sponsored by our collaboration partners. Here we're trying to combine with, as I mentioned before, checkpoint inhibitors. In the melanoma, we're combining with Keytruda, a PD-1 checkpoint inhibitor. In the peritoneal trial, we're combining with a PD-L1 checkpoint inhibitor, durvalumab from AstraZeneca. In mesothelioma, we're combining with chemotherapy. And in prostate cancer, we're combining with a dendritic cell vaccine. And we believe this combination program allows us to test which drugs our oncolytic virus works most effectively with. We recently had data in our melanoma trial from the first 6 patients. So I'll show you some of those results. We had 1 very interesting case example in these first 6 patients. This was a patient that had been treated with both Yervoy and Keytruda previously and progressed. The patient then entered our trial after progressing on Keytruda, looking like this, had a lesion on his leg. After only 3 injections of our virus, by week 3, there was a clear visual regression of the tumor. And by week 9, only 2 infusions of pembrolizumab or Keytruda, all signs of the cancer was gone. So in only 9 weeks, we got a complete response, which is very encouraging. And bear in mind, these are advanced patients, heavily pretreated, refractory to several target drug modalities. So it's very rare that you see this type of response. It's only 1 patient, but of course, very encouraging. To summarize what we saw in these first 6 patients, first, safety is of course important. We had no safety concerns reported with a combination with Keytruda, and the trial is continuing as planned. We see no added toxicity on top of the checkpoint inhibitor. Of course, we are trying to stimulate the immune system, so we need to demonstrate that we're activating an immune response. Indeed, we see innate immune activations in all the patients. They get a fever as a response to the virus. They have increase of cytokines in circulation, which we want to see. Subsequently, you want an adaptive immune activation, i.e. stimulation of T cells, recognizing the tumor. Indeed, in the patients we analyzed so far, 3 out of 4 had an increase in tumor-infiltrating T cells. In 2 out of 4, we were able to demonstrate that these T cells were specific for the patient's tumor, i.e. they're not recognizing the virus, they're actually recognizing the tumor. This is a very important mechanistic proof. And in one of the patients, we also saw an abscopal immune response. By that we mean a T cell infiltration into a non-injected lesion. So we inject one site, and we see an immune response in a different one, which is indicative of a systemic anti-tumor immune response. And as I mentioned before, we had 1 patient with a complete response. 3 others had transient regression of their injected tumors, but were withdrawn from the trial due to distant metastases. And we also see that the patient with the strongest immune response seemed to have the best effect clinically as well. Now, how we treated these patients were, the patients were enrolled in the trial. They got 3 ONCOS 102 injections, no more ONCOS after that, just standard pembrolizumab treatment. So all the ONCOS injections are early on. We did see the immune response tend to taper off over time. And we also clinically saw the tumor, these 3 patients that had some early responses later got metastases. So we believe that we're not dosing enough in this cohort. So we have decided to start a second dosing cohort where we increase from 3 dosings of ONCOS 102 to 12. So now every time the patient comes in for pembrolizumab, they will also get an injection of the virus. This is consistent with what other oncolytic virus companies are doing, consistent with our data, and we are confident that by changing to this treatment regimen, we'll be able to unleash the full potential of the immune activation of ONCOS 102. So we have closed the first cohort at a total of 9 patients, and we're now recruiting into this second cohort. And we believe we'll have data from this cohort in 12 to 18 months. The trial is running in 3 sites in the US, including Memorial Sloan Kettering. We are also developing the next generation of oncolytic viruses. These are all in preclinical development. We have 3 candidates. They're all based on the same backbone as ONCOS 102, but they have other transgenes and more sophisticated target modalities. The first virus is currently in vivo testing. The other 2 are in vitro, but they're all cloned and validated and function as they should. So to summarize, Targovax, our lead product, ONCOS 102, is an oncolytic virus with strong single-agent data, early combination responses, and 4 ongoing trials. Our TG product, which I didn't talk to you much about today, is the first ever cancer vaccine to show an immune response to a driver mutation. So this is also a very promising product, and we have an innovative pipeline of next-generation oncolytic viruses. So with that, I stop and hand over to Klaus. Thank you.